EPIC, and I'm very happy to just turn this over to Dan Tighe, who organizes our graduate scholar series.
Thank you very much, Jean. Good afternoon, everybody. Today, we're very fortunate to have Paul Muller joining us. And Paul was introduced to us through Eric Heyer. And so I will turn it over to Eric to give a formal introduction of Dr. Muller. Eric, it's all yours. You're on mute. You're on mute, Eric. You have to unmute yourself. Oh, still mute. You're still mute. In the left bottom corner, I think you can unmute yourself.
Unmute.
Okay, now you're on. Good. Oh, you're back to mute. Still muted, still muted.
Mute.
Okay, good.
Okay, I won't do anything now. I'll just continue to talk. So it's a real pleasure to introduce Paul Muller, who has agreed to talk to us today. So Paul did his undergraduate training at Stony Brook, and then was a graduate fellow at two institutions. One was NYU, Sadler School of Medical Sciences, and then the Rockefeller University. And he has had a fascinating career looking at the effect of the enteric nervous system and its development with the sympathetic nervous system. And so I was present when he gave his thesis lecture at Rockefeller, and it was just fascinating. And I had asked him if he wouldn't mind sharing his results with us today in EPIC. I also should say that Paul's father, Bob Muller, was my best friend in graduate school at Albert Einstein College of Medicine. And so I've known Paul since before Paul was Paul. And it was really wonderful to see how he has developed over the years and has really become a first-class scientist. So it's with great interest that I await Paul's discussion now on his topic. And I've succeeded in screwing up where the official title is of his topic. I can
repeat it anyway. So
okay, that would be great, Paul.
Sure.
Okay, so it's with great enthusiasm that I present Paul Muller, who's going to talk to us about the enteric nervous system and how it affects the CNS.
Yeah.
Paul?
Okay, so I just muted everyone, and then I'm gonna share. Okay. I assume everyone can see this at this point. So yeah, thanks to Eric and to Dan and to Jean for giving me this opportunity to talk. And as Eric said, my name is Paul Muller, and I'm actually currently a scientist at a biotech company in the city called Calliope, and we're studying basically the gut-brain axis and seeing how we can intervene pharmacologically there to treat a number of different diseases. So today I'm going to be talking to you about my thesis work, which was entitled Sympathy for the Microbiota, How Gut Microbes Influence Mammalian Physiology Through the Sympathetic Nervous System. So today we're going to be talking about the intestine, the mammalian intestine. And so just to give you an idea about sort of the overall architecture of the gastrointestinal tract in the mouse, if we lay this out, when food is ingested, it obviously passes through the esophagus into the stomach, then into the duodenum, and then as it progresses down into the distal intestine, into the ileum, and into the colon. And so we can think of the intestine as having sort of four basic functions. The first is detection, that whether that be sort of nutrients or microbes, as we'll come back to, or other things that are present in the lumen of the intestine. The second is affording protection against either harmful pathogens or toxins that have been ingested. The third is motility, that is moving something from one end of the gastrointestinal tract to the other. And the final is allowing for digestion of whatever is ingested, and then subsequent absorption of relevant metabolites and nutrients. And so how does the intestine actually go about achieving this? Well, it takes advantage of a number of different cell types, and the three that are pictured here are obviously not all that exist in the intestine. They're just sort of the major players at this point, and the ones that we'll be discussing a little bit today. So there's an extensive nervous system, there's the largest reservoir of immune cells in the body, and then an extensive epithelial layer. And so each of these cell subtypes has sort of unique crosstalk with the other in order to mediate each of these four distinct functions of the intestine. And so sort of how does the organization of these cells look if we look at the actual intestine? So if we take a cross section of a piece of intestine, and then we zoom in on the finger-like projections, which are the villi, and then we actually take a piece of intestine from a mouse and we make it transparent and then stain for different cell types. What you can appreciate is that stained in here in blue is a single layer of epithelial cells, which provides really the only physical barrier between the lumen of the intestine and the inside of the body. And then in green, what you see highlighted here is just one subtype of immune cell, namely macrophages. And then in red, now you can see there are neurons, and you can really appreciate sort of the dense innervation that is present at the epithelium, at the interface between the lumen of the intestine and the inside of the body. So as I said, all of these, all three of these cell types have crosstalk with one another in order to mediate each of these distinct functions of the gastrointestinal tract. But there's also another player which we haven't talked about yet, which will be sort of a major player for the remainder of the talk, and that's the microbiota. So the intestinal microbiome, we're going to refer to that in terms of mice as mice that are specific pathogen free. And today, the microbiome itself encompasses, viruses, protozoans, et cetera. But today we're just going to be referring to the microbiota, so to the microbes, the bacteria that are residing within the gastrointestinal tract. And so there are about 10 to the 13th commensal or normal microbes present in the mouse intestine. But a microbiome is, and sort of microbes are not unique just to model organisms. There is even, sort of a hipster microbiome which exists in the Gowanus canal, and I just bring this up to bring up the concept of a functional adaptation to either an area or to a model organism. So in terms of the Gowanus canal, there are distinct bacterial types strains that exist along the Gowanus canal that have adapted to process whatever toxic chemicals have been dumped in the canal over the years. And so just like the Gowanus canal, the intestine has a distinct anatomical geography in terms of the microbiota that exists there. So in the upper GI, in the duodenum, which is primarily tasked with absorption and detection of nutrients, there's very little bacterial density and diversity. And then as we move our way down into the distal intestine, into the ileum and colon, the bacterial density and diversity increases quite dramatically. So how does one actually go about studying the effects of microbes on a model organism? Well, we take advantage of a germ-free system to start off with. And so germ-free simply means that these are mice, in this case, that are born in a germ-free environment. So there are no microbes present, and they live that way until you introduce them with bacteria or other microbes that you might care about. And so the microbiome, what we've learned by using germ-free organisms over the years is that the microbiota has an effect on a number of different diseases and sort of every biological process you can imagine. Literally, if you can think of something or a disease, then likely the microbiota either has a direct or some kind of indirect effect. But in terms of how the microbiota has an effect on the nervous system that's integrated to the gut, which we're going to refer to as the enteric associated nervous system, that's been a little less studied over the years. So people have reported before that sort of within the gut, there are changes in neuronal excitability and phenotype, meaning the types of neurotransmitters and neuropeptides that are produced. And in the central nervous system, people have reported, other groups have reported that there's a major influence of the microbiota on neuropsychiatric disorders and neurodegenerative diseases. But as I said, sort of the how things affect the nervous system that's locally integrated to the gut, and then how this has an effect on physiology is sort of has been underexplored in the past. And so that was what we set out to do during the course of my thesis. The question that I had posed was really, what is the impact of the microbiota on the nervous system integrated into the gut? And then how does this translate into basic intestinal function? And that includes the immune system, because I did come from the laboratory of mucosal immunology, but also how this relates to sort of basic physiology. So because we're going to be talking about this nervous system, I just want to introduce it very briefly. So if we take a piece of mouse ileum, we make it transparent, and then we stain for nerve fibers. What you can really appreciate is sort of the dense innervation that exists not only within ileum, but all areas of the gastrointestinal tract. And so where does this innervation actually come from? It comes from two distinct sources. The first are what we call intrinsic enteric associated neurons, or enteric neurons as they're sort of classically known. And these are neurons that are have their cell bodies in the wall of the intestine. And then there's another source which comes from what we call extrinsic enteric associated neurons, and these come from either the sensory ganglion, so the nodos and dorsal ganglion, or the dorsal motor nucleus of the vagus, or the celiac superior mesenteric ganglion, and these are part of the parasympathetic and sympathetic nervous systems, respectively. So in the lab, in Daniel Masita's lab, before I sort of embarked on this, main question that we'll get to in a couple of minutes, we already had some previous evidence for microbial detection by the nervous system as it related to immune modulation in the gut. And so when I first started out in Daniel's lab, we had a story whereby we looked at a population of macrophages in the muscle layer of the intestine that we call muscularis macrophages. And we sort of wanted to understand what their function was. And what we found is that at steady state, meaning that all things are normal in the lumen of the intestine, there's a normal microbiota. We found that neurons within the gut, so intrinsic enteric associated neurons, they produce a trophic factor, colony stimulating factor 1, which allows for the recruitment and differentiation of these macrophages. And these macrophages in turn produce bone morphogenic protein 2, which then has an effect on these intrinsic neurons to modulate their activity. And this has an impact on intestinal motility in general. So the second question that we had beyond sort of the function of these macrophages during the steady state was what function might these macrophages play during the course of infection? And so what we found is that using a model of enteric infection by giving salmonella, we saw first of all that there's a population of intrinsic enteric neurons that die off during the course of infection. And this is mediated via the inflammasome, which we'll come back to a little bit later in the talk, but this is this macromolecular immune complex that can sense different sort of damage signals, and then allows for the release of pro-inflammatory cytokines, and also results in many cases in cell death. And so we saw that there was this neuronal death, but what we also found that was interesting is that there was a rapid detection by the nervous system. We still don't really understand what exactly that signal is during the course of inflammation. But what we do know is that this then activates the sympathetic nervous system, which allows for the release of norepinephrine, which acts on these same muscularis macrophages via adrenergic receptor beta 2 to change their gene expression profile towards a more tissue protective or anti-inflammatory phenotype. And so what we found in a series of subsequent studies is if we remove adrenergic signaling capabilities from these macrophages, then we find that we can increase the amount of neuronal loss that occurs during the course of infection. And on the contrary, if we come in and we exogenously activate these macrophages either via pharmacology or by activating the sympathetic nervous system, we find that we can significantly rescue the number of neurons that normally die off. And so, these two different stories involving the immune system give us an idea about, first of all, this detection, which likely happens via the epithelium in terms of commensals. This has an impact on the intrinsic nervous system. and crosstalk with the immune system to allow for an impact on motility. And then the second story involving pathogens indicates that there's detection, again, likely via the epithelium, which is then transmitted to the extrinsic enteric-associated nervous system, which can then allow for protection to be mediated, again, via the immune system. But, stemming off of those, coming off of those studies, we really had this sort of general question. What happens, how is the microbiota just having an overall impact on the nervous system that's integrated into the gut? And so again, if you take a piece of gut from an SPF mouse, specific pathogen-free mouse that has microbiota, and a germ-free mouse, what you can appreciate if you stain for nerve fibers is that from sort of a zoomed out level, and even when we zoom in on the villi, the overall structure of the nervous system appears to be relatively the same, even though in the case of germ-free mice, the villi are a little bit blunted because there can be, there are changes in epithelial turnover. So what we really wanted to do is we wanted to get a better snapshot of how the nervous system is changing. We thought we could do that by employing transcriptional profiling to look at the gene expression in neurons that are integrated into the gut. And so to do this, we decided to use this technology, which is called translating ribosomal affinity purification, or TRAP. And this version that we're using is RiboTag. And so what this allows you to do is, in a cell type specific manner, you can express ribosomes that have a little tag on them. And so you can take your tissue of choice, you can homogenize it, and then you can liberate these ribosomes in addition to all the other ribosomes that are then attached to mRNA that they're actively translating and turning it into protein. And then you can take an antibody and you can pull out those ribosomes that have that tag on them. You can then elute the mRNA and then you can sequence it. And so then this gives you a really nice snapshot of what is being actively translated at any given moment in a cell type. And this is, it gives you a little bit better resolution than the total mRNA pool because it tells you what is actively being turned into protein at that given point during the cell's life. So with this tool in hand, we use that to profile, neurons from the extrinsic and intrinsic nervous system, which led us to these two stories I'm going to talk about now. The first is showing that gut-connected sympathetic neurons we found that they're tonically inhibited by the intestinal microbiota. And the second story is that intrinsic neurons, we found are a unique population of neurons in the periphery. They're shaped by the microbiota, and we also found that they control blood glucose via the sympathetic nervous system. So getting into the first story, we first wanted to focus on the extrinsic enteric nervous system. So those neurons that are integrated into the gut but don't have their cell bodies in the wall of the intestine. And so we first wanted to sort of confirm that the relevant ganglia that we should be taking for transcriptional profiling. So in order to do this, we injected a retrograde tracer that has a fluorescent tag on it into the wall of the intestine. This gets taken up by the axons and then retrograde trafficked to the cell body. And then you can visualize neurons that are connected synaptically to the intestine. So we injected this retrograde tracer into different parts of the intestine, the upper and the lower GI. And then we found that there are indeed neurons that are connected to the intestine in the sympathetic ganglion, and then the sensory ganglion, dorsal ganglion, and nodose ganglion. So, and we also found that in the case of each of the three areas of the intestine, if you introduce three separate colors of cholera toxin, then each of those neuronal populations are non-overlapping, indicating that there's sort of unique neurons that integrate into each area of the intestine. So we then took each of these ganglion and then profiled them in SPF mice, but we also at the same time generated mice with the same genotype that were germ-free. So we could compare germ-free ganglion to an SPF ganglion. And just to make a sort of long story short in terms of the sequencing, what we found that was most interesting was that in the germ-free sympathetic ganglion, there was increased transcript of FOS. And FOS or CFOS is an immediate early gene, and this is used as an indirect readout of recent neuronal activity or plasticity. And so we confirmed that what we saw in the sequencing was indeed the case on a protein level. And so we removed the sympathetic ganglion from SPF and germ-free mice, and we found that indeed there's a significant increase in the number of CFOS positive neurons in germ-free sympathetic neurons. And we also just confirmed that this was not some kind of weird, Rockefeller germ-free facility effect, because when we took germ-free mice from another facility at Mount Sinai, we found that, again, that there was a significant sympathetic activation that we observed. And we also confirmed that this sympathetic activation is not sort of the result of the diet that the germ-free mice are on, and nor is it the result of being housed at Rockefeller, because SPF mice from another facility also display low levels of CFOS. And so, you can say that, germ-free mice, they grow up without a microbiota, so perhaps there is some kind of developmental defect that is leading to this. So what we wanted to do is see whether this was actually reversible. So we took our germ-free mice and then we colonized them with bacteria, basically introducing them to the fecal content of a normal SPF mouse that we had in-house. And what we found is that indeed you can reverse this sympathetic activation. And this is not simply the result of, it's not just the simple presence of bacteria that can allow for this inhibition of these sympathetic neurons, because when we introduce a single bacteria, so when we monocolonize mice, for example, with Akkermansia or segmented filamentous bacterium or B. frag with single strains of bacteria, we find that there is no effect on the levels of sympathetic activation. And even when we introduce a sort of larger consortium of bacteria, this oligo MM12, we also found that this was not sufficient to suppress the levels of CFOS. However, when we introduced altered Shaler flora, which is supposed to sort of best represent the different strains of bacteria that are normally present within the mouse intestine, we found that we could significantly suppress these levels of CFOS. And with another consortium of bacteria, Clostridia, which comes from Ken Hana's lab in Japan, and these are known to potently produce short-chain fatty acids, particularly butyrate, we found that we could, again, significantly suppress the levels of CFOS. So this indicates that there are specific bacteria or bacterial interactions between a number of different strains, which are allowing for this tonic inhibition to occur. So, of course, this could all just be an artifact of germ-free mice in general. So we wanted to understand whether we would see this type of activation if we took a mouse that had a microbiota from birth and we depleted it. What would we see? So we gave our SPF mice broad spectrum antibiotics in the drinking water. And what we found is that we could indeed phenocopy what we find with our germ-free mice, namely that we get this increased sympathetic CFOS. And if we break this broad spectrum antibiotic cocktail down into single antibiotics, we find that only ampicillin and vancomycin are capable of inducing this sympathetic activation. And if we give, the broad spectrum antibiotics in general, and the single antibiotics, those are all chronic and given in the drinking water. But if we give just an acute single oral gavage of a relatively broad spectrum antibiotic, streptomycin, we also find that within 24 hours, we can get this significant increase in CFOS. And this is independent of mouse strain as well, because in testing at least a couple of additional amounts that we can also induce CFOS in those that are, and these are distinct from B6 mice that we're normally working with. So, what is common between germ-free mice and antibiotics, whether they're sort of broad spectrum or a little bit more restricted? In most of these cases, there's a lack of fermenting bacteria, and these fermenting bacteria normally take fiber and then break them down into short-chain fatty acids, which are metabolites that are quite crucial for a host of sort of metabolic processes and general cell processes in the mammalian gut and in the rest of the body as well. And so when we looked at our sort of single antibiotic treated mice, we definitely saw that when giving broad spectrum antibiotics, sort of the major short chain fatty acids, acetate, propionate, and butyrate, they're all significantly suppressed, basically to below detection level. However, when we look at our single antibiotics, what was interesting is that if you'll remember, ampicillin and vancomycin were really the only antibiotics that could induce this sympathetic CFOS, and we find that they're the only antibiotics that could significantly suppress the levels of short chain fatty acids to basically below detection. And we also found this was the case with our streptomycin treated mice as well. And we know that when we give back either a full microbiota or the select consortia, we can also boost levels of short chain fatty acids in the gut. And so we then wanted to understand what happens. Can we sort of rescue, not necessarily rescue, but can we suppress this the sympathetic activation if we provide short chain fatty acids exogenously to mice that are, for example, undergoing antibiotic treatment. And we found that.
Can I ask a question? What is the implication of suppressing CFOS for those, what is that? What would it mean to the organism to have this response?
So, well, I mean, I will sort of come back to what we think what the result of the sympathetic activation is, which is slowing of motility. But it can be, for example, you could think of, if there's an infection and you have a decrease in, for example, short chain fatty acid producing bacteria, which are beneficial, then perhaps, motility slows down so that you can replenish that bacterial niche. That's one potential hypothesis for why you might want to do that. In addition, in terms of inflammation, the sympathetic nervous system we know is in the gut can be at times anti-inflammatory. So this can also be, if you have again, salmonella or another enteric infection, which is outcompeting some of these short chain fatty acid producing bacteria, then the sympathetic nervous system can come in and it's maybe a detection mechanism whereby you can whereby that there's infection, and then you can dampen inflammation.
Thank you.
Yep, you're welcome. So if we give short chain fatty acids in the drinking water, we find that we can significantly suppress the levels of CFOS again in these sympathetic neurons. And we also find that if we give a more stable form of butyrate, one of the short chain fatty acids, and this tributyrin is actually able to better reach the distal intestine where these short chain fatty acids are normally produced and primarily doing their job, we again find that we can suppress the levels of CFOS in these sympathetic neurons in both, sort of germ-free mice and also in a streptomycin-treated mouse model. So, how are sympathetic neurons getting activated? What is the circuit that we're looking at? So there are sort of three ways via which these sympathetic neurons could potentially get activated. The first is that the sympathetic neurons themselves, which are innervating the intestine, are somehow detecting the loss of the microbiota or a specific signal. The second route is that there are these neurons, which we'll come back to in the second part of the talk, which are called viscerofugal, which these are intrinsic neurons that have their cell body in the intestine and send axonal projections to the sympathetic ganglion. So they may be integrating information and activating these neurons. And the third option is that there is detection by the sensory nodose or dorsal ganglion, which then sort of integrate information and activate the sympathetic nervous system via the brain and the spinal cord. So just to cut to the chase, we confirmed that it was not sort of direct sympathetic detection, nor was it these viscerofugal neurons that were playing the role of activating the sympathetic nervous system. And so that led us to look at what role the sensory neurons, the extrinsic sensory neurons that are innervating the gut, what role they're playing. And so in order to modulate activity of these neurons, we took advantage of chemogenetics, and in this case, designer receptors exclusively activated by designer drugs or DREDs. And so what this does is this allows you to selectively express a mutated muscarinic receptor in your cell type of choice, and then it can be either of an excitatory or an inhibitory flavor, and then you can come in with a ligand that should preferentially bind to these receptors, and then you can selectively increase or decrease activity of your neuronal population of choice. And so we first set out to sort of broadly inhibit sensory neurons with the idea that potentially this might mimic the loss of some tonic signal that was coming from the microbiota. And we found that when we were silencing both the nodose and dorsal ganglion, we could sort of copy what we see when we deplete the microbiota, and that is namely we find an increase in sympathetic CFOS. And then narrowing down, if we go ahead and we more selectively inhibit just the nodose ganglion, we find that again we can phenocopy what we find in our antibiotic or germ-free mice, which is this increased sympathetic activation. And we also did a number of other experiments to show that the dorsal ganglion were not playing a role in this case. And so obviously the next question is, are these nodose neurons actually positioned to detect microbes, likely not directly detecting, but either detecting the metabolites or a signal that's being emitted from the epithelium. And in sort of previous work, the distal intestine, which is where these bacteria are, where these metabolites are, was thought to be like not highly innervated by nodosensory neurons. So we just wanted to understand for ourselves what that innervation looked like. And so we injected an AAV, which allowed for expression of a fluorescent protein in nodose neurons. So we injected this AAV directly into the nodose, waited a period of time for this fluorescent protein to fill the entire cell, including the pseudounipolar axons. And then we removed the distal intestine, so the ileum, which you see at the top, and then the colon at the bottom, made it transparent and stained for this fluorescent protein. And what you can appreciate is that there's quite dense innervation of the epithelium, which really indicates that these neurons are positioned to detect whatever signal is coming from the microbiota. So we then wanted to look centrally to understand whether there were, sort of brainstem areas that normally integrate information from the nodose, whether they're modulated when we deplete the microbiota. And so these brain areas that integrate nodose information are the nucleus tractus solitarius and the area postrema. And so indeed, what we found is that when we deplete the microbiota, we find that we get a significant increase, again, in CFOS in these relevant brainstem nuclei, which indicates that there is some sort of change that in activity that's happening in the nodose is also reflected in these brainstem nuclei. And if we come in with our, for example, exogenous treatment with short chain fatty acid, the butyrate, we find that in the case of the area postrema, we can selectively suppress the levels of CFOS there, which might indicate that the postrema itself is sort of uniquely tuned to respond to microbial signals. I should also mention that we also tested the hypothesis that short chain fatty acids might have a direct effect on the postrema and the brain in general, because the postrema, for example, is a circumventricular part of the brain, which means that it's exposed to the bloodstream. But when we did a direct intracerebroventricular administration of short chain fatty acids, this had no effect on the sympathetic CFOS that we saw in the sympathetic ganglion. So we then wanted to understand sort of whether these sympathetic neurons that we're seeing that are getting activated, are they synaptically connected to the brainstem and then eventually to the nodose. And so in order to get a handle on this, we injected a polysynaptic virus that expresses a fluorescent protein. So when I say it's polysynaptic, that means that it's moving in a retrograde manner. It's getting taken up by the axons and moving backwards in a circuit, hopping multiple synapses. So we injected this into the wall of the intestine and then just confirmed that we had fluorescent protein expression, first of all, in the sympathetic ganglion. Then we confirmed that this then jumps back into the spinal cord, which is shown here. And then we took the brain after a number of days, which should allow for significant spread of this virus into the central nervous system. We, again, cleared the brain and stained for this fluorescent protein and then used computational analysis to understand which brainstem nuclei were lighting up first. And what we found is that there are distinct areas in the brainstem that are synaptically connected to the gut via the sympathetic nervous system. And these are classically described as sympathetic premotor areas. And so these are divided into the lateral paragigantocellular nucleus, which also includes the rostroventrolateral medulla, the gigantocellular nucleus, and the raphe pallidus. And what there is to take away from here is that we found that there were sort of two distinct populations of neurons that were labeled with this fluorescent protein in these premotor areas of the brain. One was distinctly inhibitory, and the other was excitatory in terms of the neurotransmitters that they are utilizing. We also confirmed that if we waited a long enough period of time to sort of complete the circuit, that if we inject this PRV, this pseudorabies virus that expresses a fluorescent protein into the wall of the intestine, and what we do is we cut the vagus, which is the highway via which the nodose afferents and the vagal efferents will innervate the gut. So we prevent that virus from actually directly infecting those nuclei of the brain first, which would confound things. So we injected these mice with this pseudorabies virus and then waited a period of time, and we found that indeed we saw there were nodose neurons that became fluorescent positive, which indicates that there's a full circuit. It appears that goes from sort of the nodose, which is doing detection, into the brainstem, and then via the sympathetic nervous system, again, iterates the gut to control, as you'll see, motility. So, again, what are these sympathetic neurons actually doing? What is this sympathetic activation really responsible for? And so it's been reported that germ-free mice and antibiotic-treated mice have significantly delayed gastrointestinal motility, and so we just confirmed this in our own hands. And then we wanted to understand whether the sympathetic nervous system had any role in modulating this change in gastrointestinal motility. So we first gave a chemical guanethidine to allow for a chemical sympathectomy, and we found that preventing the release of the primary catecholaminergic neurotransmitter norepinephrine allowed for restoration of gastrointestinal motility to sort of normal levels. And so we then wanted to confirm that these brainstem nuclei, these premotor areas that we had identified, that we also, I should mention, that we also found were activated upon microbial depletion. We wanted to understand whether activating those would lead to changes in gastrointestinal motility. And so when we activated, first of all, this inhibitory population that we identified, we saw that there was no change in gastrointestinal motility. However, when we activated, and again, this is using this DREAD technology or chemogenetics, we found that when we activated this excitatory population that we identified, we found that we could significantly delay total gastrointestinal transit time, colonic motility, and importantly, we also found that we could significantly increase the levels of CFOS in the sympathetic ganglion, indicating that we had indeed activated those neurons by this upstream activation of these premotor areas. So just to summarize that first story, we found that the microbiota can tonically suppress the sympathetic nervous system that innervates the gut. We showed that short chain fatty acids and notosensory detection are key components that normally suppress sympathetic activity, although they are certainly not the only ones. And we showed sort of a number of other factors that might potentially play a role, but we think these are two of the key players. And finally, we showed that these sympathetic neurons that are activated upon microbial depletion, they can control gut motility, and they explain this change in motility that happens, for example, when people take broad spectrum antibiotics. So moving on to the second half of the talk, we're now going to focus in on just the intrinsic enteric associated neurons, or the enteric neurons, again, as they're classically known. And using that same transcriptional profiling technique, we really wanted to first get a handle on what enteric neurons were expressing, because although they've been known about for over 100 years, and there's been a lot of work in terms of figuring out neurochemical code, et cetera, there was very little that was known about their transcriptional profile. So we thought that this would sort of be beneficial just for the field in general. And so we first confirmed that those same ribotag mice, using that trap technology, that we could get these tagged ribosomes expressed in enteric neurons in the gut, in different areas of the gut. And then because we had our SPF and our germ-free, well, we'll get to the germ-free actually. So we took the muscle layer from these mice from different areas of the intestine, and this contains, there are two layers of enteric neurons in the gut, the myenteric and the submucosal plexus, but the myenteric contains the majority of the neurons in the intestine. So we took the myenteric plexus, and then we performed transcriptional profiling, and we then compared these neuronal populations to the extrinsic ganglion that we had isolated before. And so when you plot them on a principal coordinate axis, just to look at how sort of similar or dissimilar different populations are based on different attributes, what we find is that intrinsic or enteric neurons cluster distinctly away from extrinsic ganglion, indicating that they have a unique transcriptome, or in this case, we can call it a translatome because it's what is being actively translated. And so, what really defines an enteric neuron? We found that if we look at the gene pathways that are enriched, we find that neuropeptide signaling appears to be one of the major components that defines an enteric neuron versus, for example, another peripheral neuron. So, then in terms of how these neurons differ based on the different regions of the gut, we wanted to understand whether the gut segment that these neurons were located in had any impact on the transcriptional profile. And again, if we just do a PCA analysis, we find that these neurons from different segments of the gut do tend to cluster away from one another. And if we look at the gene pathways that are enriched, we find, for example, that in the duodenum, which is primarily tasked with nutrient absorption, there are gene pathways related, for example, to lipid metabolic processes or digestion. And in the colon, which is primarily tasked with water absorption or excretion, there are pathways that are enriched for example, excretion or response to mechanical stimuli, which indicates that these neurons are tuned to sort of be more preferentially used for motility purposes. Now, beyond the transcriptional profiling, if we just look sort of at total neuronal number, what we find is that there's a significant increase in the number of neurons as you move from the duodenum into the ileum and colon as you move down the gastrointestinal tract. And then related to those neuropeptides that I was talking about, the neuropeptide signaling, what we found is that as you move into the distal intestine, there's also an increase in the number of neuropeptide positive neurons. And there's one neuropeptide in particular that defines a specific population. This is CART, which is cocaine and amphetamine regulated transcript. And we'll come back to this neural population because we found that it was the one that was sort of most interesting when we started to probe what these neurons were doing functionally. So now we've then moved into the question of what happens when you don't have a microbiota, what kind of changes occur within the enteric nervous system. And so, just in terms of neuronal number, what we found is that there is a significant decrease in the number of neurons, preferentially in the ileum. There is a trend in the colon, although it was not significant. And when we do our transcriptional profiling and we sort of compare all of the different enteric neuron populations that we had isolated between SPF and germ-free, what we found that was interesting is that the ileum and colon enteric neurons from germ-free mice tended to cluster around the duodenum samples, both from SPF and germ-free mice. And so if you recall from the beginning of the talk, the duodenum has very little bacterial density and very little diversity. And so it appears that the microbiota is having a distinct effect because those distal areas of the gut, which now lack a microbiota, are now tending to look more duodenum-like. And we found that what was changing specifically in the ileum and colon, what was less expressed in germ-free mice in the ileum and colon were pathways for neuropeptide signaling and neuropeptide genes in general. And if we went ahead and we actually quantified the number of neuropeptide positive neurons, and here we're just going to keep coming back to CART because it is the neuropeptide that we're going to focus most closely on, we found that the number of neuropeptide positive neurons was significantly decreased in both the ileum and colon. So again, as we had described in the first story, things can always be attributed. Maybe there's something odd about germ-free mice that can explain this phenotype. So we colonized our germ-free mice and then looked at, first of all, the total neuronal number and found that we could significantly rescue this in the ileum and the colon. There was no change, but again, there was no decrease sort of to begin with. And if we look at our neuropeptide numbers, we found that we could significantly rescue the number of neuropeptide positive neurons in the ileum and in the colon. And again, we also wanted to understand if we take an SPF mouse and we remove its microbiota, will we see similar changes in terms of neuronal number and neuropeptides? And that is indeed the case. When we deplete the microbiota, we find that there's a significant decrease in the number of enteric neurons, and most potently, again, in the ileum. We also know that this is reversible. So if we take our mice off of the antibiotics and put them back onto Splenda, which is our control, we find that the number of neurons will go back to normal, which indicates that this is not a permanent loss, and there is some sort of precursor that exists in the enteric nervous system, which is quite interesting, which is replenishing those neurons which were previously lost. And we also showed that, again, in the ileum and colon in antibiotic-treated mice, there was a significant decrease in the number of neuropeptide positive neurons, in this case, specifically CART. So we first wanted to understand how are these neurons actually being lost? And as I said, at the very beginning of the talk, we had found that enteric neurons express distinct components of the inflammasome pathway. And the inflammasome pathway is this sort of macromolecular complex, which allows for detection of damage signals. And then this then activates caspases, which can do a couple of different things. they can first convert propeptides into actual functional inflammatory cytokines. So you get production of IL-1 beta and IL-18, but you also have processing of gasdermins, which can then insert themselves into the membrane of the cell, and this leads to a programmed form of cell death, which is referred to as pyroptosis. And so we hypothesized that perhaps the inflammasome might be involved in cell death in the case of the loss of the microbiota, as was seen with our during the infection models that I had described before. And we found that this was indeed the case. So if we look at this in knockout mice for distinct components of the inflammasome, and we also did this in a more selective manner, which I'm not showing here, by specifically removing these caspase genes in neurons selectively, we find that we can rescue. So we no longer get the neuronal death with antibiotic treatment, and we no longer get this change in neuropeptide number. So I guess the next sort of logical question for us was, well, what exactly does this mean for the mouse? And I guess sort of more importantly, we don't really have a great appreciation for what enteric neurons do in terms of just because of the fact that we haven't been able to before selectively activate them and then look at different phenotypes. There's been a lot of sort of pharmacology that's been done and a lot of ex vivo work, which has been truly informative, but there was really not much that was done in terms of using modern neuroscience tools to really manipulate the activity of enteric neurons. And so, we had looked at a few of their neuropeptides that changed levels with regards to germ-free versus SPF, but the one that appeared to give the most interesting phenotype was this cocaine and amphetamine regulated transcript positive population. And so CARD is normally is sort of classically ascribed to its function in the hypothalamus, where it's an anorexigenic neuropeptide. So it suppresses food intake when you give it in the brain or when you activate selectively those neurons in the hypothalamus. And when we looked at a sort of typical, a classically ascribed function of the enteric nervous system, when we looked at changes in motility, we found that activating selectively these CART positive neurons in the ENS did not have an effect on gastrointestinal motility. But what was very interesting was that there was a significant suppression of food intake. And so then, the next question that we had was, well, how exactly is this happening? And one potential route is that there is a change in nutrient availability. And sort of one of the most potent ways of inducing satiety is increasing levels of blood glucose. And so when we looked at the levels of blood glucose after activating these CART positive neurons, we found that there was a significant increase in blood glucose, and interestingly, there was a significant decrease in the levels of insulin in the blood. And then when we tried to sort of chemogenetically silence these neurons acutely, we didn't find an effect. But when we introduced sort of a more long-term loss of function, so when we ablated these neurons using a toxin that's expressed in an AAV to selectively deplete these CART positive neurons in the distal intestine, we found that we could reverse what we saw with this activation, which is that we get a significant decrease in blood glucose and a significant increase in insulin levels. And so how is this actually being mediated? How are these neurons doing this? So what we had found is that when we inject this DREAD, this chemogenetic tool, into the wall of the intestine, it also has a fluorescent protein that's expressed. And so you can visualize where axonal projections are actually going to. And so in the course of checking for sort of off-target effects by looking in different extrinsic ganglion where we might get some kind of retrograde trafficking, or maybe there is AAV that gets into the blood and transduces neurons there. What we found, even though we found that there was no transduction of cell bodies, what we found is that in the sympathetic ganglion that's connected to the gut, there was a significant amount of innervation that was presumably coming from the gut itself. And so that led us to the idea that these CART positive neurons are likely a subpopulation of visceral fugal neurons. And if you remember from the first part of the story, we had described these visceral viscerofugal neurons as being neurons whose cell bodies are in the intestine, and they send their axonal projections to the sympathetic ganglion. And so we wanted to know whether the gut itself was connected to glucoregulatory organs. So the liver and the pancreas, for example, the liver for gluconeogenesis and the pancreas for production of insulin and glucagon. And we want to understand whether these organs are synaptically connected to the intestine via the sympathetic ganglion. So we took this, again, this pseudorabies virus, which is polysynaptic. We used a couple of different colors, and we injected one color into the liver, the other color into the pancreas. We first confirmed that indeed we did see transduction of neurons in the sympathetic ganglion that come from both organs. And then when we looked in the intestine, what we found is that, interesting, in the ileum, we found that there were, only in the ileum, there were two distinct populations of fluorescent positive neurons, indicating that these neurons in the ileum are synaptically connected both to the liver and to the pancreas. So they can potentially control these neurons via sympathetic neurons, these organs via sympathetic neurons. And importantly, what we found is that when we activated these CART positive enteric neurons, we could get a significant increase in the levels of again, sympathetic CFOS, indicating that indeed these visceral fugal neurons can control sympathetic activity and likely control these neurons which are innervating both the liver and the pancreas. So then how does this kind of all tie into antibiotic treated mice? When we look at viscerofugal distribution, what we know is that the distal intestine, again, has the highest percentage of viscerofugal neurons. And what we found is that in line with the overall reduction in intrinsic neurons after antibiotic treatment, we also found there was a significant reduction in the number of viscerofugal positive neurons. So likely we are reducing the amount of excitatory input that is going to the sympathetic ganglion. And we also noted that in mice that were treated with antibiotics, and this is all, the same is true for germ-free mice, there's a significant reduction in blood glucose. And when we use those mice, which are either a total knockout or a neuronal specific knockout for components of the inflammasome pathway, we find that there's no change in the levels of blood glucose. And so, how are these changes in blood glucose actually being mediated? Well, there can either be control of, glucoregulatory hormones such as insulin, or there can actually be, gluconeogenesis that's happening. And so when we looked at, for example, the levels of insulin in our antibiotic treated mice, we found that there was no difference in levels of insulin in the blood. Again, there were also no differences in glucagon, but when we probe the ability of mice to perform gluconeogenesis by giving pyruvate, which is a precursor to glucose, which is then processed in the liver, we found that mice that are treated with antibiotics and also germ-free mice had a significantly blunted ability to produce glucose after getting a bolus of pyruvate. And again, if we looked at this same ability to generate glucose in mice that can no longer lose enteric neurons after when they're on antibiotic treatment, we find that there's no change in their gluconeogenic capacity. And then just to come back to these CART positive neurons, what we find is that when we use that same technique to ablate those neurons in the distal intestine, we find that we can phenocopy what we see, meaning that we can blunt the ability of those mice to mobilize glucose after getting a pyruvate bolus. Okay, so just to summarize the second part, what we found is that enteric neurons are unique. They're adapted to their different gut regions, and they're impacted by the microbiota. We showed that inflammasome signaling leads to enteric neuronal loss during the course of antibiotic treatment. We showed that this CART positive population of enteric neurons is changed by the microbiota, and it can control blood glucose via the sympathetic nervous system. And finally, we showed that loss of the microbiota blunts gluconeogenesis, and this may potentially be happening through a reduction in viscerofugal signaling that's coming from the gut. And so just sort of the overall conclusions from my thesis, summing up the whole talk, what we've shown is that sympathetic activation, when there's detection of a pathogen in the lumen of the intestine, This can lead to an enhanced ability for macrophages to protect neurons in the gut through adrenergic receptor signaling. We showed that the commensal microbiota can tonically suppress gut sympathetic activation. We showed that detection of a pathogen or dysbiosis by intrinsic neurons via the inflammasome leads to neuronal loss. Again, we showed that enteric neurons, they're unique and they're microbially shaped, and they're a different population as compared to other peripheral neurons. And finally, we showed that, again, the microbiota can program neuropeptide positive intrinsic neurons to control blood glucose via the sympathetic nervous system. And so, overall, what we had found is that, the sympathetic nervous system is sort of this nexus whereby changes in the microbiota can be integrated, and then this can mediate changes either in the immune system or just in sort of general intestinal and extraintestinal physiology. And with that, yeah, I should thank Daniel and like all my collaborators in the lab during the course of my thesis, and obviously all the funding that I had before I came to a relatively well-funded biotech. And yeah, and again, thanks to Jean, to Dan, and to Eric for asking me to give this talk today. So thank you.
Thank you very much, Paul. A fascinating presentation and a lot to chew on there. I really enjoyed the very systematic approach that you took in tackling these complicated problems. Now, I think that in your introduction, you teased us a little bit by saying that changes in microbiota could be associated with neurodegenerative diseases and possibly neuropsychiatric diseases. Where in your research would you establish that link, if it's available at all?
So I don't know if my work would directly link to that, but we just touched upon short chain fatty acids, and actually short chain fatty acids are not fantastic microbial metabolites in terms of their ability to get into the periphery. For example, butyrate is mostly absorbed by the colonic epithelium. Propionate is also relatively well absorbed. So acetate makes its way a little bit into the bloodstream. But that being said, there are also this sort of like multitude of microbial metabolites that make their way into the bloodstream and can permeate the blood-brain barrier. So those are, I think, able to have an effect on the circumventricular organs, and then this can have an effect on different neuronal nuclei. In addition to that, there are also all these effects that your microbes are going to have on, so for example, on the immune system. So if you have dysbiosis, you're going to have an increased inflammatory environment in the gut. Perhaps this leads to increased circulating cytokines, which can also have an effect on, it's been implicated, people who have depression have, it's been shown to have increased circulating levels of cytokines. There are also, in terms of, microbes can have all these effects on release of, neuropeptides that come from enteroendocrine cells. And so these can also have an effect humorally, via the circulation that then gets to the brain. But in terms of my specific work, it was really like, there's a ton of effort being put into the brain at the moment, but we were really sort of interested to just kind of dig into the locally integrated nervous system, because that was sort of underappreciated, what kind of effect the microbiota would have there.
Great, thank you very much. Any other questions? Please unmute yourself if you. Yes, Fran.
Yes, I have a question. I was just thinking as you were speaking about what a remarkably collective enterprise this is. And at the picture you showed us at the end with, a large number of people all standing together who have been helping in this work, there obviously must be a lot of coordination going on. And then there must be issues that come up with, like, whose name comes first if you publish an article, or even supposing that you discover some miraculous new drug that can be patented and everybody would become a millionaire. Do you have to sign some, is that all negotiated in advance? Like Columbia gets this much share and you get that much share, and how do these working arrangements get settled? I'm just interested in that.
So I guess there are two separate questions there. In terms of, like authorship, et cetera, like these are complicated issues, and there's no sort of set way that things are done. I think that should be definitely changed, but usually it's the person who conceives of the project and puts in the most legwork and writes the paper, that person ends up being first. But usually, I mean, in general, the last author is going to be the one who retains most of that intellectual property. And then in terms of what does the institute is going to own everything that we at Rockefeller, they would own everything that we would produce, and then if there was a patent or something that would come out, there's a sort of set split in terms of how much you would get as an inventor and then how much the university would take. And then there is the issue of whether the professor has to agree that the student or the postdoc be included on that patent or on the invention in general. But now going into industry, it's a very different setup. There's no, sort of everything is just generally associated with the company. So there's the overall understanding that whatever I do here belongs to Calliope, belongs to the company. We still do have the opportunity to publish, and hopefully we're going to do that in sort of the coming year. But there's a little less in terms of whatever you do here, it's going to benefit the company and it's going to benefit everyone here, because eventually we'll become public. So there's less to worry about in terms of what kind of split you're going to get, because there's not really that opportunity.
Great. Robin, yes?
I wonder if I want to improve my microbiome to reduce weight gain and inflation, is there anything I can do, and can taking probiotics help?
Okay, maybe I'll tackle the second part first. So I think probiotics can help for sure. The difficulty there is that you have to continuously take them. So sort of once you stop taking your lactobacillus or whatever it is, and they're at quite high doses, they're going to be flushed out kind of immediately, because they don't get a chance to really establish a niche in the gut. In terms of how can you improve your gut health? I'm not a physician, so I can't give any recommendation there. The only thing that I know that is definitely beneficial is increase your fiber intake. In general, that is overall good, whether it's by eating fiber-rich foods or supplementing with fiber. That is good for reducing inflammation, improving motility, et cetera, just general GI function. And that is definitely via production of short chain fatty acids and having an effect on the microbiota. So that would be my only true recommendation.
Okay, but probiotics, is there a particular brand or type you recommend?
No, I don't really have much experience with
But
probiotics.
you mentioned a particular one, lactobacillus or something. Yeah.
Oh, I think lactobacillus is in general included in most probiotic yogurts, et cetera. I think it's supposed to be generally calming for the gastrointestinal tract, but I don't know, it doesn't work for everyone, but it's the most commonly used.
And if a yogurt has live bacteria, that means it's automatically probiotic, even if it doesn't say it on the thing, or does it not mean that? Because the probiotic brands aren't very good.
I mean, if they're claiming probiotic, they usually, it's usually because they're including a specific strain, a specific culture in there that they think is beneficial.
Oh, so you're stuck with the ones claiming probiotic, even if they're not.
Yeah, I think so.
Thank you.
Yeah, you're welcome.
I had I was intrigued by this increasing and decreasing of neuronal activity. Is it just the activity that's changing it, or is there an actual change in number of neurons that are present? because there's, we in my
Oh,
age
yeah.
group, we grew up thinking that, you don't make new, you get what you have what you got, and that's it. You're stuck with it. So, and the second, I'll ask both questions. Second is the interaction with something like the stress response. Is that is there a similar kind of response in the microbiota that there is in your own gut system to let's say increase cortisone or whatever else happens during the stress response?
Okay, so so in terms of, okay, so there is no change in neuronal number in terms of like sympathetic neurons or other peripheral sensory populations of neurons. The thing that does change is the enteric nervous system, and this was, so for a long time the dogma was that in the enteric nervous system, what you have is what you will always have, and that's it. And so if things die off, then that's it. But before we published this work, there was a group that claimed to have found a population of stem cells that were Nestin positive in the mouse GI tract that they saw could make new neurons. During the course of I didn't see that there was any change in this Nestin positive population. I don't know if that is truly what is allowing for replenishment, but clearly we see that the ENS is plastic, which it was not thought to be before in mice. I don't know if this is the case in humans. It's I don't really know exactly how you would go about figuring that out at this point. But in terms of where that population is coming from, either there's another distinct stem cell population that we haven't identified, or it has been reported before, for example, under inflammatory conditions, that glia can sort of transdifferentiate into enteric neurons. Then in terms of the microbiota response to stress, there must be a change that happens, because we know, for example, there's work from Sarkis Mazmanian's group at Caltech. It's a preprint right now, but they've shown that if you modulate the enteric nervous system, so if you change motility, this has an effect on the types of bacteria that you have in the GI tract, and whether you increase or decrease. So it's not entirely surprising. So obviously, like, physiological stress can make you constipated, can make you have diarrhea, etc. So it can change motility, and other things, like levels of inflammation can change, and these can all have an impact on microbial community. So yes, I think sort of like indirectly they're adapting to changes in the host, to change where what niche they're in, or how many bacteria there are, etc. What metabolites they're producing. Yeah.
So the chronic use of antibiotics in our food supply is pretty, we really have no idea what the long range implications are of feeding all our chickens and pigs and cows and everything else in the world with antibiotics.
There is definitely that component. How much we are actually ingesting ourselves, I don't know, but you bring up a good point, that just in general, in terms of medicine, the prescription use of antibiotics is so, for example, I was talking with a friend who is a surgeon, and we were just discussing the fact that people who have bronchitis, etc., like clearly viral infections, they're prescribed antibiotics as sort of like a calming factor. And I know personally, when I had my gallbladder moved like 6 years ago, and they put me on broad spectrum antibiotics, so I was completely wiped out in terms of microbiota. And there's no plan in most hospitals for giving you even a probiotic, again, something to make sure that you are colonizing yourself again with the correct microbes. There are some people that pay attention to it, but I think across the board, you get an antibiotic and nobody's going to pay attention to what happens afterwards. They just want to treat the infection, and not, there's not much attention that's paid to the after effects of depleting either the majority of the microbiome or a large portion of it. So I think it's definitely, I mean, it definitely has major implications in terms of human health, and it's, I think, something that people are becoming more aware of.
Any other questions? I have another one, and I don't know if you can answer it, but
I'll
you
try.
mentioned that you work in for a private company now.
Yeah.
So what is a commercial angle that's of interest to the private company to sponsor your research?
Wait, can you repeat? What is a commercial?
Why is this? Why did the private company hire you? What are they?
Okay. Well, I mean, I can answer this. So actually, I don't work on the microbiota here at all. In fact, there's very little that is done here directly. We do work sort of with microbial metabolites, but I have to say that the field of using the microbiota to treat human health is truly in its like infancy. There's so much people know very little about sort of single strains of bacteria. Very not to mention like how they interact with one another and how they interact with like the host, et cetera. So a lot of that stuff I think is kind of, I mean, it's very hot right now, but I don't know if there's a lot of truth to it. I think we're just at the very beginning stage. There's a lot of work that needs to be done. So I was hired because of my sort of gut brain experience, my experience with immunology and sort of more specifically with the enteric nervous system, because so I'm sort of tasked here with sort of probing the enteric nervous system more on a basic science level to figure out, trying to parse out what can we do from the gut via the enteric nervous system that can impact physiology in general, inflammation, and then seeing, if we can actually if we can find, targets on neurons in the gut that we can then go after with pharmacology and some tricks to keep things, restricted to the gut, and see if we can, use that to treat, I don't know, diabetes or treat Crohn's and colitis, IBD, things like this. So that, they brought me in really for the nervous system component. The microbiology stuff, maybe down the line, if we become really successful, maybe we'll bring that back.
Thank you.
We'll bring that into the fold. Yeah.
I thought that the interaction between the pancreas, the spleen, the liver, and the gut that you showed is everything is broken down into such specialties when we look at the human being, and there's just such a lack of appreciation of the complexity and the interaction of the whole body and all its systems, and then the way that it interacts with the external world. I thought your work, that was very interesting, that part of your work.
Yeah, and I think, I mean, that is one of the things that we are starting to, that I'm starting to appreciate more during the course of this job, is that, really the ability to use the gut as a way to sort of bypass, the central nervous system, for example, to control a lot of basic physiology is a really attractive and super interesting avenue of research. So yeah, I think there's only more that we will discover.
So what do you think about bariatric surgery? What is that? What are the implications of something like that?
So bariatric surgery, I'm trying to remember. So
I think it's the stomach that they go after, right?
Yeah.
Where do they do
Mostly the stomach.
Yeah.
I mean, there are like three different, there's like the sort of gastric band where they decrease the stomach size.
Yeah.
There is there's like, varying levels of gastric bypass where they, cut at the pyloric sphincter and they connect that to like the jejunum or the ileum. So people think there are these hypotheses that there are changes in gut peptide expression. So you get a really significant increase, for example, in GLP-1, which we know now, has like profound effects on satiety and blood levels, et cetera. There's also the fact that because you're bypassing the sort of more absorptive part of the gut, you're also, preventing yourself from taking up, the majority of fats and glucose, et cetera, that you're ingesting. But at the same time, it may very well be that now you're also activating some really potent signaling pathways because you're introducing, for example, these enteric neurons to levels of nutrients that they wouldn't otherwise see. So there may be, I had thought about that a little bit, whether there's a role for these, visceral fugal neurons in mediating changes in blood glucose after bariatric surgery, because I know, for example, Roux-en-Y is actually like, you can essentially cure diabetes via that procedure. And so it's really unclear to people how that's done. And I personally think, it can't be solely explained by the fact that you have whopping levels of GLP-1 that are produced, because there are companies that are based on just giving that peptide and you don't have the same dramatic effects.
Duct tape on the mouth. That's
Yeah.
the solution.
Yeah, that also works.
All right, any other questions? If not, I think we're going to conclude here. I want to thank you, Paul. It was a real fascinating presentation. Clap, and I want to thank Eric for introducing you and bringing you to our attention. It was really a pleasure to.
This was a lot of fun. Thank you very much for giving me the opportunity to talk. And maybe I will see a number of you in the future. But yeah, thanks for having me. I appreciate it.
Thank you.
Thank you.
And our next session